Hello and welcome to the lecture on Genetic Material Based Diagnosis for Viruses, Part 1, Unraveling the Molecular Secrets. Have you ever wondered how doctors figure out which virus is making you sick? In this video series, we'll embark on a fascinating journey into the world of viral diagnosis. We'll be uncovering the clever tools scientists use to detect and understand different viruses, like the one behind COVID-19. This isn't just about scientific curiosity, though. Accurately identifying viruses quickly is a critical weapon in our fight against infectious diseases. By understanding the specific culprit, doctors can choose the most effective treatments and public health officials can take targeted measures to control outbreaks. Throughout this series, we will delve into the exciting world of diagnostic techniques. We'll explore the challenges scientists face from differentiating between similar viruses to keeping up with the constantly evolving pathogens. We'll also discover groundbreaking innovations that are revolutionizing the way we diagnose viral infections. Unveiling the enemy. Why we need laboratory virus diagnosis. Imagine fighting a battle blindfolded. That is what treating viral infections can be like without accurate diagnosis. But the good news is that modern labs provide the necessary tools for a clearer picture, leading to significantly better patient care. It helps avoid unnecessary tests and antibiotics. When we know exactly what virus we are dealing with, we can avoid unnecessary tests. Antibiotics work only against bacteria and not viruses. Therefore, antibiotics can be avoided, and thus the problem of antibiotic resistance can also be avoided. It allows for quicker management and informed prognosis. Accurate diagnosis means swifter treatment. Knowing the culprit allows for quicker initiation of targeted therapy. It allows for targeted interventions. For example, early detection of herpes virus during pregnancy allows for a C-section to protect the newborn. Informed decisions on pregnancy continuation. For example, knowing the presence of virus like rubella empowers the informed decisions about pregnancy continuation. Accurate diagnosis is the foundation for effective antiviral treatment as well. It helps in formulating a targeted therapy. Gone are the days of generic treatments. Modern antiviral drugs work best against specific viruses. Knowing the enemy allows for the targeted therapy with maximum effectiveness. Chronic infection management also becomes possible. For long-term battles with viruses like HIV or hepatitis B, accurate diagnosis is crucial for treatment. Treatment decisions can also be made through accurate diagnosis. Choosing the right antiviral medication based on the specific viral strain is helpful. Monitoring response is also possible to adjust medications. Tracking viral load to assess treatment effectiveness and identify potential drug resistance. Adjusting medication strategies is also useful. Early detection of drug resistance allows for timely adjustments to medication regimes, ensuring continued treatment success. The fight against viruses extends far beyond the walls of a doctor's office. Modern labs play a critical role in protecting public health by providing the data that drives crucial public health measures. Blood transfusions are life-saving, but they can also unknowingly transmit viruses. Lab testing ensures the safety of blood supply by screening for HIV and hepatitis. These are just examples of viruses that can be transmitted through blood. Screening ensures that donated blood is free of these pathogens. When someone tests positive for a sexually transmitted virus, contact tracing allows public health officials to identify and test potential contacts. This helps prevent further spread of the virus. It, Education and awareness campaigns can be targeted to higher risk populations. Early detection of a virus outbreak within a hospital is vital for rapid intervention. Implementing isolation protocols and disinfection measures quickly can prevent further infections. Protecting vulnerable populations is especially important for patients with weakened immune systems. Every year, flu strains can mutate. Lab results from around the world help scientists identify new influenza strains. This allows for the development of vaccines that target the most prevalent strains. By knowing which strains are circulating, public health officials can prioritize vaccination for at-risk populations. Mosquitoes can transmit a variety of viruses. Lab testing identifies infected mosquitoes. This allows for targeted mosquito control measures in affected areas. Early detection can prevent large-scale outbreaks by tracking the spread of arboviruses. Containing In case of a new or exotic disease outbreak, lab results are crucial in containing them. Rapid diagnosis allows for isolation and treatment. Implementing containment strategies is possible to check further spread. We have explored how modern labs empower better patient care and public health measures, but the fight against viruses doesn't stop there. We need to be constantly vigilant, tracking, and understanding these ever evolving threats. This is where surveillance comes in. Modern surveillance programs allow us to track the prevalence, monitor the spread and prevalence of known viruses within a community. It helps understand the impact of different viruses on public health, prioritizing interventions for those with the greatest burden. This data fuels informed public health decisions, leading to more effective control measures. Targeted interventions like vaccination programs or public awareness campaigns can be designed, develop, or adapt. It helps develop or adapt vaccination strategies based on the most prevalent viral threats. While we have made significant strides, the world of virus is vast and constantly evolving. Surveillance is critical for detecting new epidemics. It is important for identifying new outbreaks of unknown or previously rare viruses before they become widespread. It helps in unveiling hidden diseases, discovering new diseases altogether, often caused by viruses we haven't encountered before. It helps in connecting the dots, uncovering previously unknown links between viruses and specific diseases. Believe it or not, over 90% of human viruses were unknown just after World War II. Surveillance is the key to staying ahead of this hidden threat. By continuously monitoring viral activity, we can anticipate potential outbreaks, develop new diagnostic tools, and ultimately protect ourselves from the ever-evolving world of viruses. Revolution in viral diagnosis, from clinical picture to laboratory precision. Why are traditional methods unreliable? Symptoms like fever, fatigue, or a cough can be caused by a variety of viruses. It is like trying to solve a mystery with just a few blurry clues. This uncertainty can lead to delays in proper treatment and unnecessary suffering. But today, the landscape of viral diagnosis has undergone a revolution. Modern laboratories are playing a vital role in this transformation, thanks to the powerful trio of advancements. First, molecular technologies. These are the game changers. They allow us to directly detect the presence of viral genetic material, leading to faster and more accurate diagnosis. 2. Antiviral advancements. Gone are the days of one size fits all treatments. With the rise of targeted antiviral therapies, identifying the specific virus causing an illness is crucial. It's like having the right key to unlock the correct door. This targeted approach leads to more effective treatment and reduces the risk of antibiotic overuse, which can contribute to antibiotic resistance. Furthermore, there is an increased demand for information. Clinicians are no longer satisfied with just knowing you have a virus. They need detailed information to make informed decisions. This is where modern diagnostics come in. We can now measure the viral load, which tells us how much of the virus is present, giving the sense of disease severity. Additionally, we can analyze the viral sequence, identify the specific strain of the virus. This is crucial for understanding how it might behave and respond to treatment. Finally, these tests can also detect antiviral resistance, allowing doctors to choose the most effective medication right from the start. We have established that traditional diagnosis has its limitations and modern labs are crucial for accurate viral identification. But what exactly makes these modern labs so powerful? Let us explore some key benefits. Modern labs are equipped to process a large number of samples simultaneously. This translates to faster testing times for patients, leading to reduced wait times. No more waiting for weeks for results. Doctors can make treatment decisions sooner, potentially improving patient outcomes. Time is the essence when it comes to viral infections. Modern labs deliver results significantly faster than traditional methods. This allows for swifter treatment initiation. The quicker we identify the culprit, the sooner we can target it with the right treatment. Early intervention can minimize the severity of illness and potential complications. Modern labs aren't isolated entities. They foster a collaborative environment where lab technicians and clinicians work hand in hand. This close communication facilitates more informed diagnosis. Clinicians can leverage the lab's expertise to interpret results in the context of a patient's specific symptoms and medical history. Lab results guide treatment decisions, ensuring the most effective approach for each patient. What makes an ideal diagnostic method? Here are the key criteria we strive for. Speed. Time is of the essence when dealing with viruses. An ideal test delivers results quickly. Simplicity. The best tests are user-friendly and don't require extensive expertise to administer. Sensitivity. A sensitive test accurately detects even low levels of the virus, minimizing the chance of a false negative missing virus when it is present. Specificity. A specific test only reacts to the target virus, reducing the chance of a false positive, mistakenly identifying a different virus. Cost. An ideal test is affordable and accessible to a wide range of patients and healthcare systems. Just like there are a variety of viruses, there are several techniques to detect them. Viral diagnostics encompass a spectrum of techniques. They are broadly categorized into two approaches. One, the direct detection of virions, viral antigens, or viral nucleic acids in tissues, secretions, or excretions. Second, indirect detection of the host immune response, that is, antibody-based detection. Introduction to viral genetic material based diagnosis. At the core of every virus lies its genetic material, which can take various forms and structures. Understanding the diverse nature of viral genomes is essential for researchers, healthcare professionals, and public health officials in their efforts to combat viral diseases, develop effective diagnostics, and gain insights into the evolution and behavior of these microscopic entities. The most commonly used system of virus classification was developed by Nobel Prize winning biologist David Baltimore in the early 1970s. Baltimore classification scheme groups viruses according to how the mRNA is produced during the replicative cycle of the virus. Group 1 viruses contain double-stranded DNA as their genome. Example for this is a smallpox virus. The mRNA is produced by transcription in much the same way as with cellular DNA. Group 2 viruses have single-stranded DNA as their genetic material. An example for this group includes the parvovirus. They convert their single-stranded genomes into a double-stranded DNA intermediate before transcription to mRNA can occur. Group 3 viruses have double-stranded RNA as their genetic material. An example for this is the rotavirus. Their strands separate, and one of them is used as a template for the generation of mRNA using the RNA-dependent RNA polymerase encoded by the virus. The group 4 viruses have single-stranded RNA as their genetic material. An example for this group are the coronaviruses. The group 4 viruses have single-stranded RNA with a positive polarity. Positive polarity means the genomic RNA can serve directly as mRNA. Intermediates of double-stranded RNA, called replicative intermediates, are made in the process of copying genomic RNA. Multiple full-length RNA strands of negative polarity complementary to the positive stranded genomic RNA are formed from these intermediates, which may serve as templates for the production of RNA with positive polarity, including both full-length genomic RNA and shorter viral mRNAs. Group 5 viruses have single-stranded RNA as their genome. Example for this is the measles virus. The single-stranded RNA genome has negative polarity, meaning that their sequence is complementary to the mRNA. As with group 4 viruses, the double-stranded RNA intermediates are used to make copies of the genome and produce mRNA. In this case, the negative stranded genome can be converted directly to mRNA. Additionally, full-length positive RNA strands are made to serve as templates for the production of negative stranded genome. Group 6 viruses have single-stranded RNA as their genetic material. Example for this is the HIV virus. These viruses have deployed two copies of single-stranded RNA genomes that must be converted using the enzyme reverse transcriptase to double-stranded DNA. The double-stranded DNA is then transported to the nucleus of the host cell and inserted into the host genome. Then mRNA can be produced by transcription of the viral DNA that was integrated into the host genome. Group 7 viruses have double-stranded DNA as their genetic material. Example for this is the hepatitis B virus. The group 6 viruses have partial double-stranded DNA genomes and makes single-stranded RNA intermediates that act as mRNA but are also converted back to double-stranded DNA genomes by reverse transcriptase necessary for genome replication. There can be structural variations in viral genomes. The genome may be circular or linear. An additional variation in viral genome is seen in certain RNA viruses, such as the influenza virus. Here, instead of existing as a single molecule, it may be segmented, existing in several pieces, each of which may encode a separate protein. In some plant viruses, the segments may be present in separate particles. So in order for the replication to occur, a number of virions need to co-infect the cell, thereby complementing each other. That is, they must have multipartite genomes. Double-stranded RNA is always present in the segmented form. Now let us discuss the impact of genomic variation and genomic diversity of viruses on viral diagnosis. Viruses constantly adapt and change, making them the fastest evolving entities in biology. But what fuels this rapid evolution? Here's a breakdown of the key factors. Short replication time. Viruses multiply rapidly within host cells. This rapid replication cycle creates numerous opportunities for mutation to occur during each copying process. It is like having a recipe constantly being rewritten by hand with a higher chance of errors creeping in each time. High number of offspring. A single viral infection can produce millions of virus particles. The vast number of offspring increases the likelihood that some will harbor mutations, creating a diverse viral population. Several mechanisms drive this rapid evolution. Those are random mutation, recombination, reassortment, gene amplification, and reduction. Random mutation of viral genomes. The mutation rate is the frequency of new mutations in a single gene, nucleotide sequence, or organism over time. Viral genomes display a higher mutation rate than cellular organisms. In some viruses, such as the retroviruses or the human immunodeficiency virus, the mutation rates may be as high as 10 to the power of minus 3 to 10 to the power of minus 4 per incorporated nucleotide. Viral polymerases are more error prone than cellular polymerases, with the least accurate being viral RNA dependent RNA polymerases. Moreover, most RNA viruses or cytoplasmic DNA viruses can't use the cellular proofreading machinery, which is functional for nuclear double-stranded DNA, and they generally don't encode this function. Genetic recombination is the exchange of part of genome between two genetic entities. There are two kinds of recombinations, self-recombination and recombination with the host. Self-recombination occurs when two closely related viral genomes recombine by homologous crossing over. This happens for DNA viruses and is very common in prokaryotic viruses. Some RNA viruses also evolve by recombination. It is common for retroviruses. These recombination events can be an evolutionary advantage for the virus when they help it evade host immune defenses. For example, by changing surface protein antigenicity. Recombination with host or other organism occurs when a viral genome acquires sequences from a cellular organism. This allows the virus to capture cellular genes and hijack their function, which can provide an evolutionary advantage for the virus. This event is commonly seen in large double-stranded DNA viruses like the pox viridae, and it is used in biotechnology to produce vaccines. Protist and algal nuclear cytoplasmic large DNA viruses may have acquired many genes from bacteria that are the prey of the eukaryotic host cell. Reassortment occurs in segmented viruses only. Reassortment event occurs when two similar segmented viruses exchange part of their genomes during a cell co-infection. This way, segmented viruses can modify quickly their antigens, creating a new strain that can evade the previously established immune protection of a population. This has been particularly well studied for influenza virus since reassortment is a major event which gives rise to new flu pandemics. Gene amplification is a process that produces extra copies of the same gene in a viral genome. In Poxviridae, host genes that have been included in the viral genome by recombination can be subject to amplification. The host gene will be multiplicated on the viral genome. More copies increase the probability of beneficial mutations. After acquisition of a beneficial mutation, these viruses can lose the non-beneficial copies. Now let us look into the challenges in diagnostic assays due to viral genetic diversity. The high mutation rates and genetic diversity inherent in viral populations pose a significant challenge for the design and reliability of diagnostic assays. Many diagnostic tests rely on the detection of specific genetic sequences or markers within the viral genome. However, as the virus replicates and accumulates mutations, these target regions can become altered, leading to a mismatch between the test design and the actual genetic composition of the circulating viral strains. This can result in reduced sensitivity or even complete failure of the diagnostic test to detect the virus. Designing effective primers and probes for diagnostic assays is critical, but it is made increasingly complex by the genetic diversity of viruses. Researchers must carefully select target regions that are conserved across multiple viral variants to ensure broad coverage and sensitivity. However, as the virus continues to evolve, these conserved regions can become less reliable, leading to the emergence of viral escape mutants that evade detection. Continual monitoring and optimization of primer and probe designs are necessary to maintain the effectiveness of diagnostic tests. The presence of viral quasi-species, a diverse population of closely related viral variants, can further complicate diagnostic testing. Within a single infection, there may be a heterogeneous mix of viral strains, each with its own unique genetic signature. Diagnostic assays designed to detect a specific variant may miss or underrepresent other variants present in the sample, leading to inaccurate results or an incomplete picture of the viral diversity. This challenge is particularly acute in settings where multiple viral strains co-circulate, such as during outbreaks or in regions with high genetic diversity. The genetic diversity of viruses also poses challenges for effective disease surveillance and monitoring. Diagnostic tests that fail to detect emerging viral variants can hinder the ability to track the spread and evolution of these pathogens, limiting our understanding of their epidemiology and hampering efforts to implement appropriate public health interventions. Maintaining robust and adaptable diagnostic capabilities is crucial for early detection and effective response to viral disease outbreaks, particularly in the face of rapidly spreading disease. Now let us discuss the important methods for genetic material extraction. Now let us discuss the importance of extracting genetic material in the diagnostic workflow. Extracting genetic material of viruses is a critical step in the diagnostic workflow. The extracted genetic material, that is DNA or RNA, serves as a foundation for subsequent analysis. By analyzing the extracted genetic material, we can precisely identify the specific virus causing the illness. Beyond just identification, extracted genetic material allows us to further characterize the virus. We can analyze its strain, the potential mutations it carries, and even its relationship to other viruses. Without efficient extraction, the entire diagnostic process can fail. Inaccurate results, impurities, or insufficient genetic material can lead to misleading or inconclusive test results. Imagine analyzing a contaminated fingerprint sample. You might end up with the wrong suspect. Without a clear picture of the culprit, accurate diagnosis becomes an elusive goal. Techniques for extraction of viral genetic material. The viral genetic material can be extracted from a range of samples like the environmental samples, animal tissues, or cell culture based samples. The traditional method utilizes organic solvents like phenol and chloroform. One can also add chaotropic salts to disrupt the cell membrane and separate DNA from cellular components. The addition of organic solvents like phenol and chloroform induces phase separation, creating distinct layers within the extraction mixture. Nucleic acids preferentially partition into the aqueous phase, while proteins and other contaminants remain in the organic phase. DNA can sometimes remain at the interface between the two phases. Following phase separation, the nucleic acids can be selectively precipitated from the aqueous phase by the addition of isopropanol or ethanol. The precipitation step removes residual salts and contaminants, yielding a crude nucleic acid pellet. The pellet is then washed with ethanol to remove any remaining impurities before being resuspended in an appropriate buffer, such as a TE buffer, for further analysis or downstream applications. For RNA extraction, additional steps are often included to preserve RNA integrity and remove the contaminating genomic DNA. This may involve treating the crude RNA extract with RNase inhibitors to prevent degradation by endogenous RNases present in the sample. DNase treatment can also be employed to degrade any contaminating DNA, ensuring the purity of the RNA sample. Finally, both DNA and RNA extracts are typically quantified using spectrophotometric methods such as UV absorbance fluorometry or quantitative PCR. Additionally, the quality of nucleic acid extract can be assessed by measuring the A260 by A280 ratio or the A260 by A230 ratio for RNA, which indicate the purity of the sample by assessing the presence of contaminants such as proteins or organic solvents. While the method is quite effective, organic extraction can be time-consuming, laborious, and requires careful handling of hazardous chemicals. 2. Column-based methods. This popular technique uses available spin columns containing specialized filtration materials. The sample is sliced, broken open, and then passed through the column. The column selectively binds DNA or RNA while the other cellular components are washed away. This method is faster and safer than organic extraction, making it ideal for high throughput applications. Magnetic bead-based methods. This innovative approach utilizes magnetic beads coated with molecules that specifically bind to DNA or RNA. The sample is mixed with beads and the target molecules are attached to the bead surfaces. By applying a magnet, the beads and bound genetic material are separated from the rest of the sample. Magnetic bead-based methods are fast, simple, and allow automation, making them well suited for large-scale diagnosis. Factors influencing the extraction efficiency and purity of viral genetic materials. Sample type. Variability in sample matrix, example blood, saliva, tissue, can affect extraction efficiency. Presence of inhibitors, example heme, humic acids, may interfere with nucleic acid extraction. Viral load. Higher viral loads typically result in greater nucleic acid yield and improved extraction efficiency, whereas low viral loads may require specialized extraction protocols or increased sensitivity in downstream assays. Storage conditions. Proper storage of samples at appropriate temperatures, example -80°, preserves nucleic acid integrity. Degradation due to freeze-thaw cycles or prolonged storage can compromise extraction efficiency. Lysis conditions. Optimization of lysis buffer composition, pH, and temperature influences cell membrane disruption and release of nucleic acids. Incomplete lysis of cells can lead to reduced extraction efficiency and contamination with cellular debris. Choice of the extraction method. Different extraction methods, example, organic extraction, column-based, magnetic bead-based, offer varying degrees of efficiency and purity. Selection of the most suitable method depends on sample characteristics, desired yield, and downstream applications. Inhibitor removal. Addition of specialized reagents, example, proteinase K, cheotropic salts, help to mitigate the effects of inhibitors present in complex samples. Inhibitor removal steps are crucial for improving extraction efficiency and reduce fake false negative results. Elution volume and concentration. Optimization of elution volume and concentration methods influence the final yield and purity of extracted nucleic acids. Balancing yield and purity is essential to ensure optimal performance in downstream assays. Contamination control. Stringent adherence to sterile techniques and contamination prevention measures minimizes the risk of cross-contamination during extraction. Regular monitoring of extraction reagents and equipment for potential sources of contamination is imperative. Quality of extraction kits and reagents. Selection of high quality extraction kits and reagents with validated performance characteristics ensure reliable and reproducible results. Batch to batch variability and quality control measures should be considered when choosing extraction products. Operator skill and experience. Proper training and proficiency in extraction techniques are critical for achieving consistent and reliable results. Attention to detail, adherence to standardized protocols, and troubleshooting skills contribute to successful nucleic acid extraction. Now, let us discuss some MCQs on this aspect. What challenge does viral genomic variation pose in assay design? A. Decreased diagnostic sensitivity. B. Simplified primer and probe design. C. Enhanced assay specificity. D. Increased complexity and potential for false negative results. The answer is D. Increased complexity and potential for false negative results. Which type of nucleic acid is found in the genetic material of retroviruses? DNA, RNA, both DNA and RNA, neither DNA nor RNA. Answer is B. RNA. Which structural variation is not commonly observed in viral genomes? A. Linear, B. Circular, C. Helical, D. Segmented. Answer is C. Helical. Which of the following is not a key role of viral diagnosis in disease management? A. Identifying the causative agent of infection. B. Monitoring treatment efficacy. C. Improving patient communication. D. Implementing infection control measures. Answer is C. Improving patient communication. Why is it important to consider genetic diversity in viral diagnosis? A. To limit diagnostic accuracy. B. To ensure uniform assay performance. C. To avoid challenges in assay design. D. To accurately detect diverse viral strains and quasi-species. Answer is D. To accurately detect diverse viral strains and quasi-species. Viral diagnosis plays a vital role in individual patient care only. D. Public health measures solely. C. Both individual patient care and public health measures. D. Neither individual patient care nor public health measures. Answer is C. Both individual patient care and public health measures. References: Fenner and White's Medical Virology. Thank you for your patient listening.